Greetings everyone, my name
is Alexander James Edwards.
is Albert Hines.
It's Hong Jet Lee, and
Welcome
welcome to our
to our
PowerPoint
presentation
presentation
on
on
the
the FSH
FSH
cell
cell
signaling
signaling
pathway. Before we go into the specifics, first let's define what a cell signaling pathway is, which is a series of chemical reactions in which a group of molecules in a cell work together to control a cell's function, such as cell division or cell death. It starts with a hormone that binds to the cell's receptor, thus receiving a signal which then activates the next molecule, and the process repeats until the last molecule gets activated, and the cell function carries out. Settling that, now we define what an FSH is, which stands for follicle stimulating hormone. FSH is important in sexual development and reproduction, core components of human life. It's released by the pituitary gland and stimulates ovarian follicles, a sac containing egg cells. It plays a key role in fetal development, menstruation, puberty, and oogenesis, but how is it triggered? It happens through a feedback system called hypothalamic pituitary gonadal axis, where a hormone known as GnRH stimulates the release of FSH that will bind to the gonads. By then, it controls the amount of GnRH released through another hormone, which inhibits the production of GnRH.
Now that we've defined the basics, we can get to our main point. This is the FSH cell signaling pathway. It starts with the hormone itself, FSH, or sometimes called folliotropin, which is a combination of follicle-stimulating hormone beta and glycoprotein hormone alpha. The hormone then binds to the follicle-stimulating hormone receptor, activating a downstream signaling chain reaction through multiple different pathways. Right now, I will explain the pathway to our lab. Most of the protein in this pathway is all about metabolism, proliferation, cell survival, growth, and angiogenesis. First, it goes through TSC2 and then go through RAT and to the protein AKT1, which all of them regulates the protein mTOR. mTOR itself controls them into different set of pathways like RPS6 that was activated by the encoding of X6K through phosphorylation of RPS6BK1 and 2, which promotes protein synthesis, cell growth, and cell proliferation. EIF4EBP1, which regulates protein synthesis, while also activating SGK1, which regulates cell survival, neuronal excitability, and renal sodium excretion.
I'm Alexander James Edwards, and I'm doing the middle pathway for FSH signaling. So, let me start off with PRKCA, which is involved in the regulation of lipids and glucose metabolism, and is a component of the intracellular signal transduction mechanism. For example, regulating CREB1 through phosphorylation, and HIST3H3 and MAPK14 indirectly through unknown means. PRKCA, do the little flip. Moving on, CREB1 is in charge of regulating CYP19A1, this one here, which instructs the making of enzymes that convert androgens to estrogens, steroidogenesis down here. And CREB1 also facilitates AREG through phosphorylation that promotes the growth of AREG that promotes the growth of normal epithelial cells. It also inhibits the growth of certain aggressive carcinoma cells, and also has developed other functions in mammary glands, oocytes, and bone tissue development. Further go on, CREB1 also regulates BDNF, which promotes cell survival, differentiation, and death, or apoptosis. Moving on, the right-way pathway is involved in the control of a variety of cellular processes through proliferation, differentiation, motility, and adhesion, such as through CRC. Many of the things are quite unknown on how each of them lead on to the other, but the majority of the proteins within this pathway are in charge of the listed before. So RAF1 right here from SRC provides instruction for the control of proliferation, differentiation, and migration, and apoptosis to the proteins like MAPK1 down here and MAPK3, which can phosphorylate into PRKCA down here. Plays in many different cellular processes such as cell adhesion, cell transformation, cell cycle checkpoint, and cell volume control. If you see a little funny thing which I found while doing some research on the shapes, MAPK3 looks sort of like a squid. Quite nice.
Okay, so for mutations, we are going to focus on the FSH receptor as it plays an important role in fertility. The mutations in the FSHR can make it more or less sensitive, or even block it completely from binding with the FSH beta. This can drastically decrease the production of eggs or sperms, overall affecting the reproductive growth. For women, it can lead to conditions like primary ovarian insufficiency, where ovaries fail to produce eggs properly, and for men, it can lead to testicular failure. For treatments, we have IVM, where immature eggs are collected and matured in the lab. Another option is the molecular chaperone therapy, which involves proteins that can help FSH receptors work better. Or we can use intracytoplasmic sperm injections, where a sperm is directly injected into an egg. If these do not work, eggs or sperms can be freezed for future use.
Here are some of the citations that we used in the presentation.
That's it. Thank you for watching.